Hello, it's Alpesh Doshi here again from IVF London, bringing you yet another episode of Inside the Laboratory and diversifying or demystifying the embryology elements of IVF. Today I want to speak a bit about genetic testing in the embryology lab. So some of you may have heard of PGD or PGS. What does this actually mean? The terminology stands for pre-implantation genetic diagnosis or pre-implantation genetic testing. So pre-implantation genetic diagnosis is when you have couples who are actually carrying a genetic disorder within the family. So you may have heard of some couples actually carrying a genetic abnormality within their family or within their genes, as we call it. Some of these abnormalities are cystic fibrosis, beta thalassemia, and sometimes even cancers such as breast cancer, which can flow through generations, and because there is this faulty gene that gets passed over. So patients who actually come for genetic diagnosis want to try and have a healthy child free from the genetic disorder that is being questioned. So what do we do in IVF to help couples like these? So we first have to continue doing IVF as normal. We would then perform a procedure called embryo biopsy. What we are doing with embryo biopsy is we're taking a small sample of their embryos. So of course, we know that these patients may not be infertile, but they need to have IVF because we need to create their embryos in the laboratory and then do the genetic testing. So once these embryos have made it to a point of development into the laboratory, we would perform what we call the embryo biopsy. I'm going to show you a video of the embryo biopsy itself, and here we are. We're just going to rewind this. So this is the embryo at the blastocyst stage, and we have what we call a hatching blastocyst here. We're then taking some cells in a pipette, and we are then using a laser beam to cut the cells very gently and dissociating the cells away from the embryo. So you may already see there that we're applying the laser and peeling the cells away from the embryo. There we are. So that was quite noticeable, and there's a small clump of cells that we have taken from the main embryo. This small clump of cells would then be sent for genetic analysis, and the report would be available to us within a week or so. And then we would know whether this embryo is affected or free from the genetic disorder that we're talking about. So that's a biopsy at the trophectoderm or blastocyst stage. You can also do the biopsy at what we call the cleavage stage of the embryo, when the embryo is around 6 to 8 cells in development. So this is a beautiful 8-cell embryo there, and you can see what we're doing is we're taking a couple of cells out of the embryo and sending it for genetic analysis. So this is what embryo biopsy would be about, and we should be telling you a bit more in the other episodes about how the genetic testing is carried out. So hopefully this has given you a bit of insight about what we do for genetic screening. It is a huge subject, and we're going to try and do this in various smaller episodes. So today we showed you what embryo biopsy looks like. The next discussion we're going to have is when you get the genetic results, how do you prepare the patients with the information, and how you prepare them for the actual transfer as well. Thank you so much.